Good afternoon. Welcome everyone to TRI Translation Seminar Series. And before we begin, on behalf of TRI, I pay our respect to the Turrbal and Jagra peoples, the traditional custodians of the land and waters of the Brisbane area. We recognize their continuing connection to land, water and culture. We pay respect to elders past, present and emerging. We acknowledge the stories, traditions and living cultures of Aboriginal and Torres Strait Islander peoples and commit to fostering a culture of learning from and working with First Nation peoples in the spirit of reconciliation and access to justice. My name is Irina Buckle. I'm a early career researcher here at Mata Research at TRI, and it's my pleasure to welcome two excellent speakers today, our very own TRI's Fiona Simpson, Associate Professor Fiona Simpson, and Dr. Tracey Brown, and they will take us on a bit of a journey. Exciting journey. So our first speaker, Dr. Tracey Brown, who joined Telix in February 2020 as the Global Director of Clinical Operations. Over the last 25 years, Tracey has founded and acted as Chief Scientific Officer and Chief Development Officer in several global biotech companies and worked with European and US biotech companies to lead product development, taking products from conception all the way through to registration. And through this process, Tracey has developed broad-ranging experiences in manufacture of chemical and biological therapeutics, development and implementation of preclinical and clinical development plans, regulatory affairs via interaction with international regulatory agencies and management of clinical trials all the way from phase 1 to phase 3. Tracey obtained her doctoral degree in biochemistry and molecular biology from Monash University and is a graduate of the Australian Institute of Company Directors. Welcome, Tracey. And our very own Associate Professor Fiona Simpson, familiar to many of you, has spent over 25 years working on creation of therapeutics for a wide range of cancers. And today we are going to hear about their journey to commercialize a potential treatment for adenoid cystic carcinoma. So thank you very much both joining us today, and we look forward to your talks. Thank you, Tracey. Take it away.
Thank you, Irina, for the introduction. I appreciate it. Hi, I'm started at Telix 2 years ago as the Global Vice President of Clinical Operations, but over the last year, I've actually changed my role. I'm now the Global Senior Vice President of Product Portfolio Management. What that basically means, I've got a bit of laryngitis, so I apologise. I hope you can hear me. And what that basically means is I run the product development for Telix, and also, so that's up to the point of regulatory approval, and also beyond that. So I have a commercial team as well as a product development team working with me. And what I wanted to do today is really introduce you to Telix, and so you can understand how we interact with non-commercial organisations. So if you have a product in mind or product concept that you think Telix might be interested in, please do not hesitate to reach out to me. Because we are an ASX listed company, I have to put up this disclaimer. So that basically means anything that I say to you today is not enticing you to subscribe or buy shares in the company, and everything that I'm saying is non-confidential. And so it's just part of being ASX. I need to put that up there. A little bit of background about Telix before I go into an overview of our non-commercial collaborations. So the thesis behind the Telix company is to take radiopharmaceuticals to see cancer and then to treat it. And it's such an exciting approach. After working in oncology for the last 25 years with oncology products, I made a conscious decision to move to Telix because of how I see we could add increased benefit to patients by so easily taking a targeted moiety and changing radioisotopes. So what Telix does, as you'd appreciate, within a tumour, it's highly heterogeneous. You'll have quite a number of different subpopulations. And those subpopulations are characterised by specific proteins or markers within or on the tumour cell surface. And that's what Telix leverages. So we have a research program or product development program targeting 4 main epitopes or targets within cancer. That is prostate membrane-specific antigen, LAT1, carbonic anhydrase 9, and CD66. There are 4 main targets, but we're constantly looking for new targets, and so we're constantly inviting people to come forward if you think there's something else we could use as a means of payloading or delivering our radioisotopes to tumour cells. So we have our payload or our targeting moiety, be it small molecule or an antibody. We put a chelator on it and then add on radioactivity. So in order to image, and that's systemically then administered to the patient. So in order to image, we use an isotope such as gallium-68 or zirconium-89, and we inject into the patient. And I don't know if you can see my pointer here, probably, I hope you can. Anyway, if you have a look at then you image the patient with a SPECT or PET-CT. And because of the isotope that we use and the specificity and sensitivity of the targeting moiety, we can see disseminated cancer. So in the bottom right-hand corner, this is a really good example of a prostate cancer patient who's been imaged with our gallium-68 PSMA targeted product. And you can see by all the small black dots where the cancer's progressed and the state of disease, which is highly advanced. Depending on the product, within 1 day to 2 weeks, when the half-life of the radioisotope from the imaging procedure has died down, we take the same targeting moiety and we just switch out the radioisotope. That means we'll come in with the therapeutic, be it 177 lutetium or 131 iodine or 225 actinium. And these are therapeutic isotopes, so they go directly to the same place that the imaging agent went, but this time they give out a beta isotope or an alpha isotope that has cytotoxic effects. And it basically goes in and kills the cancer that we've already seen. So that is the premise of Telix products. Radiopharmaceuticals is an area that's really advancing very quickly. Just in the time that I've been in radiopharmaceuticals, a number of companies are going up exponentially. Telix is one of the leaders at this point. We last year dosed over 12,000 patients. We also in December was able to get our first imaging product approved by the FDA. And we have 17 clinical trials going, but in 7 different indications, which shows the diversity and the wide application of our products within Telix. What differentiates us from other companies is the amazing manufacturing and supply logistics distribution network that we have. Our main competitors will maybe have 1 or 2 manufacturing sites where they use that to deliver all around the world. What we have tried to do within Telix is to build a manufacturing footprint in 11 countries with over 80 countries within our distribution network. So when we have head offices in Indianapolis, Liège and Brussels and Geneva and also Melbourne and Kyoto in Japan. And so between our head offices and our manufacturing supply teams, we're able to supply radioisotopes to the entire world, which for us, a company that was only founded 3 years ago in Australia is quite an achievement. This is an overview of where our products are treating cancer. We have our brain cancer glioma program. We also have extensive trials going in breast cancer, lung, bone marrow conditioning, bladder cancer, kidney, and prostate. With our portfolio, when I took over my role, I decided to try and expand the portfolio in a way that we could take our products and treat each part of the cancer patient's journey. So as you know, majority of patients will first be treated with surgical procedures. Then patients are imaged and you get your therapeutic. So within each of our 4 targets, we have products that cover the surgical aspect, also the imaging as well as the therapeutic that we're developing. This is our research program. So we have an active research program as well as product development. And we are looking at alpha therapies, which are the next generation in radiopharmaceuticals. Beta emitters, which most of the products are at the moment, are highly effective, but they have a large penetration range, which can also result in toxicities, but are fantastic for large tumours. While the alpha therapies have a smaller penetration range within the tissue, but they don't have the toxicity and very short half-lives. So they can go in and get to the micrometastases, knock it out quickly without enhancing toxicity for the patient. We also have setting the groundwork now for our cancer immunotherapy combinations. We have tumour microenvironments. So if you have any targets or research ideas within any of these streams, please reach out to us. where we're looking at combining our products with the standard of care treatments, in looking at the tumour microenvironment in particular. One of my personal favourites is artificial intelligence, which is so cool because we get all the images from these patients, and through developing algorithms, we're able to look at these images and then design software that can predict how a patient's going to respond to certain therapies or how long it's going to take for them to progress. Still in early days, but we have a team under the direction of Dr. Michael Whitcroft who's doing an amazing job in all of these research areas. And also we have a radio-guided surgery program where we believe in only taking out the cancerous tissue and leaving as much of the healthy tissue as possible, especially in a disease such as prostate cancer that has major quality of life benefits for the patient. So why collaborate with non-commercial organisations? It needs to be a win-win for both parties. Now, over my last 20 years, coming from an academic background, this is what and being on the commercial side, this is what I've noticed. The pros for a company is most definitely a win-win because within the university or institute, you have highly dedicated teams of smart people who generally come up with amazing ideas. So that's the first major advantage of working with an institute such as TRI. Also, excellent infrastructure. So you're able to provide really scientifically stringent data that can accelerate the development of a product. So Telix, for example, we only have our own in-house manufacturing. We outsource absolutely everything else. And we work very closely with academic institutes who have developed models and validated models to work with them in order to accelerate our development programs. Within the institutes and academic institutions, subject-specific experts. We leverage experts all the time from non-commercial organisations. And if we can, in fact, work with the institute to obtain government funding, that's a really good way of getting our product development done quickly and cheaper. And there's a win-win for both Telix as well as the institute that we work with. Now, there's always a few detractors. The cons that I've noticed, and I'd love to hear what some of you guys think during question time, is there tends to be a lack of understanding around product development timelines and milestones. And that's absolutely nothing wrong with that. It's just when someone approaches us with a preclinical asset and says, oh, you know, in 3.5 years' time, you'll be able to have this approved on the market. We do get that. So it's often an exchange of information, which I thoroughly enjoy, to take, having been there myself as an academic, taking someone through the whole drug development process to come up with collaborative and agreed milestones and timelines. Also, another con that or disadvantage has really been around research being found on cool ideas, which are fantastic, but sometimes you can't find a commercial focus for that. And we can't work with the team who's a great idea, but will actually end up without having a product we can sell, or where there's a need for it within the market or within the clinic. This is something I've actually struggled with a lot in my career, is balancing the need to publish versus patent. It's getting better, but we really, as an academic, you still need to publish your work in order to maintain your profile and also to get the funding. And so it means sometimes you have to, I suppose, sacrifice that commercial application to your research work, because normally you need to patent, wait for at least a year before you can publish. And that can be quite difficult, depending on the work. If you can put in provisional patent, but it is a bit of a balancing act that you have to play with. Then also, as I said before, finding focused research that you can actually put into product-focused outcomes. That's another issue too, because everyone gets carried away with the research program. It becomes a baby in a lot of ways, and it doesn't always translate into a commercial finding. As an ASX listed company, every project we have has to generate not only scientific and clinical benefit, but also financial benefit for shareholders. So we have to take that into consideration. So what is the process within Telix? I'm sorry, I'll go through this very quickly. So we have an organisation approach Telix, or in the case of Fiona, we think she's wonderful. We've reached out to her, and we get the scientific or clinical data provided to Telix. At that point, it goes to a product evaluation committee. We look at the data. So that committee within Telix is myself, our Chief Medical Officer, and also Michael Whitcroft and our business development group. And we will go through certain, we look at it as per what you would submit to the FDA under an IND type focus. We probably say no in about at the current rate, about 60% of the opportunities presented to us. And if we say yes, it then goes to the next stage. We provide a short list of questions that go back to the party providing the product concept. Look at those. We also then do a commercial and clinical competitive environment analysis. And if all of this lines up, Telix would then provide a term sheet to the organisation, and that basically goes through the ground rules of how we're going to interact. So be it through how much funding we'll give you, what sort of milestones and timelines we have to collaborate and work towards. Then we put a research plan in place where it's a very strong collaboration between the academic institute and also the Telix product development team. At that point, we then sign the contract. Now, really quickly, this is just a snapshot of the commercial collaborations and non-commercial collaborations we have going globally. And I'll just quickly show you in Australia who we work with. We work with University of Sydney, with Peter Mac, with ANSTO, numerous hospitals, with OzHealth, Genesis Care, and that can be from anything from preclinical all the way through to clinical assets. So we have an active collaboration process, and we're always happy if we can expand upon that footprint of collaboration. So thank you.
Hi, Tracey, that was absolutely fantastic. I'm hoping just to swap to. Is this showing? Yes. So Tracey and I actually met probably over 15 years ago when we were both doing contract work for Merck KGaA and talking at one of their seminars. So I just want to really quickly give you some principles of what we've learned dealing with companies for the first time, and certainly through our initial set. So a common misconception is that if you collaborate with industry, it will pay for your research. It won't. Research and academic research have grants for a reason because they're discovery based. Whereas when you're working with a company, their priority is their product. It's to sell, it's to get it through to patients, registered, clinical practice, and there's no messing around. And so they don't exist to in any way fund your cool ideas because that's not a commercial interest. Now, there's a really good resource for all researchers. I've put the link up at the top. It's the Corbell Academia Industry Collaboration Best Practices. It's a beautiful set of guidelines for academic researchers and how and why they should interact with industry. Here you can see some of the main advantages: complementing competencies, knowledge, technological advances, obtaining access to resources that are specific to the partners, broadening the scope, isn't it? Hold on, I just need two.
It's not shared.
How do I get this to? Hold on, I just need to share this. Where's the share? There we go. Yeah. Okay, got it. Right. So that's the misconception. This is the link to the academic industry guide. So access to resources, which is has been and is an important one for our laboratory. It broadens the scope of your ideas and your innovation. And I find that the companies really help with that innovative thinking because they have a different way of thinking often than researchers do, and also critical mass. And the companies and their connections are also very good at bringing together experts in the field and people that you might not know that they introduce you to. And I'm going to give you an example of that. So how do you go about identifying potential partners? You need to look through the companies to see what your potential partner profile is, create a proposition that fits them, keeping in mind that they don't do research. They manufacture and commercialise and register therapeutic goods, certainly in our field, and then reach out to prospective partners. So I have a table here. I'll go through some examples. So what are the functions of those relationships? We can generate and share knowledge. And that can be as simple as testing something, and an important outcome for that can be if you find a side effect or a drug interaction that might be important for them going forward to clinical trials. That information in itself can be a useful thing for academics to feed to pharmaceutical companies. You get access to the company expertise, sometimes high-end, high-throughput core facilities and vice versa. Consumables and materials subject to MTA, they have really good drugs, and getting those supplied by the company can be really good for an academic laboratory. Joint publications with higher impact because you've gone through that whole usefulness process, and expand your professional network, enhanced quality of reproducibility and research outcomes because it really needs to be correct, spot on and statistical if you're working with company partners. More attractive terms for obtaining licenses, generation of IP, personnel exchange. For instance, Jason was in this morning as a PhD student looking for a company placement for his 6-month sabbatical. I'll probably talk to Tracey about that later. And more opportunities for co-funding. So company letters of support, co-grant applications, other such things. So there's a lot of advantages to working with the companies. I'm not going to take you through our data. I'm just going to take you through the process of how we find relationships. And this isn't about that place where it's already a contractual working relationship. This is about how you build a relationship and how you get started. One of the things that's happened recently is that the phase 3 data from lutetium therapy for prostate cancer has come out, and it was a little disappointing. So although there is a rise in efficacy for PSMA lutetium in prostate cancer, the clinicians weren't terribly impressed by that rise in efficacy from the Dutch groups. And one of the issues with that for the hospitals on a really practical basis, and this is why you have to talk to companies, researchers and hospitals, is Ewan Walpole wasn't very happy that his cancer budgets would be spent changing toilets and things to deal with radioactive patients and the infrastructure that they need to do lutetium therapy in exchange for that level of improvement in outcomes. So what we'd like to do is test J591 as an actual therapy and add in J591 with the lutetium, and with the various improvements in adjuvants, see that if you can get a really large improvement in efficacy that even Ewan Walpole will be happy to spend his budgets changing the toilets for. So as you can see, that's just a really kind of odd example. We just started chasing J591. It's ended up being multiple conversations about other things that we won't talk about here as well. But one of the things I want to say to the students is Chris and I met years ago, Tracey and I met years ago, Jason Lewis, we were already chatting to about his paper. And it's amazing as you work through the years how all these people kind of end up in the same circle, and that the connections between them sometimes just all fall into place at the right time so that you can get something quite big done quite quickly. So my big advice to all the young ones is just talk to people. Even if they're not interested at that time, meeting them, knowing who they are, where they're going and what they do is really useful. Okay, questions? I'll put Tracey too.
Yeah, I can kick off. Both of you spoken at length about the sort of pros and cons between academia and research, academic research and industry, and what sort of, I know you kind of touched on that, Fiona, already, to advise that you can offer to early mid-career researchers who are sort of fairly junior in their career, appreciating the translational potential of their work, but at the same time seeking some sort of industry engagement. And as you know, not labeling it as a hobby project, because quite often that's what it is.
Absolutely. So I think for early to mid-career researchers, the advantage of talking to the companies is to find out how the companies think and what directions they would take that research and what they see value in. Because what we see value in as researchers is not the same as what the companies see values in. Would you agree with that, Tracey?
I would agree, yes.
And also what the clinicians see value in is sometimes slightly in between those things or removed from them. So you actually have to talk to multiple groups of people. So I think the advantage for the young ones is if you want to really take what you're doing, like if you're happy doing academic research, that's absolutely fine. And it's what I did for years. If you really want to change your academic research into something useful on the planet that people use immediately and not in the future, then you have to talk to the clinicians and you have to talk to the companies, because the only way you can do that is to take it through those two sets of people, and they have to guide you. Like, they need to say, these are the steps that I would take with this to make it a commercially viable product. If it's not commercially viable, it'll never really reach the patients. You agree with that, Tracey?
I think that's a really good point. So, I was an associate professor at Monash Uni for 10 years before I started my own first biotech company. And for me, what caused that transition was I had been involved in a clinical trial and realised that as a researcher, although I was doing some really interesting, great work, I wasn't actually addressing what I needed to address to get my research ideas into man, which is ultimately for me, I'm a translational scientist, so that's what I wanted. And so by being able to do exactly what Fiona said, and that is to talk to clinicians, also to get help from regulators. So a large part of, because we didn't have time, is you need to work so closely with the FDA, EMA, PMDA and CDE in China to understand what they need in a product to get it approved and into patients. So it's almost like you have to first work out what your product has to address in the clinic, then make sure it's commercially viable, and then just work backwards to ensure you have all of the quality, all of the right steps in place. When I interview people for roles in Telix, I say, tell me how you would do the following thing. And 9 out of 10 of the translational people I speak to say, well, of course, you start at the end and work backwards. And that's exactly what you have to do. And I think that would be my advice. Instead of being so tied up in all of the research ideas, which is fine, but try and think to the next step of what does this product concept have to be and what does it have to achieve to get into man.
I would add to that once you've talked to the companies, you also have to talk to the clinicians. Yeah. Because they have to be prepared to use it and use it in the setting that you would have to use it.
Exactly.
So if you've got a drug that has to be injected every single day into a patient, you're going to meet some resistance to that. Yeah.
A good example is the example that Fiona gave on the prostate cancer PSMA product, where the data has come out. So at the moment, patients have to be treated 6 times with really high levels of radioactivity, and the therapeutic benefit wasn't as great as you would really need to justify uptake of that product. So as soon as we saw that, Telix really readdressed some of our clinical studies to the point where we're now putting in our antibody only twice as a therapy, and the doses of radioactivity are lower because our antibodies stick around for longer versus small molecules, so you don't need to administer 6 times. And it's that sort of advantage that can make or break a product for the clinician, for the patients, and also for the company that's developing the product as well. I did have a question from someone asking about immunogenicity of our antibodies.
Hi, Tracey, do you think you have problems with antibody immunogenicity?
Yeah.
Ranjini, feel free to unmute.
It's Professor Ranjini Thomas, Tracey.
Well, we, of our antibodies that are currently in man, we have humanized our antibodies. And so we don't have immunogenicity. We have one chimeric imaging antibody. And when we looked at the anti-chimeric antibody generation, it's between 1% to 2%. So that we haven't experienced that issue.
Yeah, and I guess you're only using the antibodies for a short period.
That's right. And so especially in the imaging and also really low doses too. So we've in all of our, but as part of any clinical study associated with an antibody for imaging or therapeutic, you do have to take samples and do your hacker testing or your human antibody development assays. And that's routinely part of any of our preclinical and also clinical studies.
I had another question, and that is, how do you go about finding your academic partners? Or is it by word of mouth, or do people come to you, or do you find that easy or difficult?
So a large part of my role is to read all the literature. And so you know what it's like, you get to know who the experts are and you approach them. And because of my academic background, I have a lot of connections, like working with Fiona previously, and you just get to know the right people to go to. But it's really sticking in with the literature and staying current with that'll help you.
So the only, sorry, John Hooper here has a question.
I'm not sure if Tracey can hear this, but
I'll repeat it, John.
Is Telix interested in different payloads like fluorescent probes or cytotoxic?
Is Telix interested in different payloads like cytotoxic or fluorescent?
Cytotoxic, not at the moment. Definitely we're radiopharmaceutically focused, but we have got a product we're doing a lot of radiosurgical guidance work. So we actually do have a product that has a fluorescent payload as well as a radioactive. So we use our gallium-68 PSMA small molecule imaging agent, and we have a fluorescent component to that as well. And which means that we're able to image the tumour and then, of course, use fluorescence to work out the margins of dissection. So we're also looking at that in renal cancer at the moment with Oxford University, and in non-small cell lung cancer with Oxford as well. So we are interested. If you have anything like that, please approach us, because all of the portfolio, I'm building it on 3 pillars: surgical, diagnostic, and therapeutic.
Tracey, I just sent her a LinkedIn.
Tracey, he just sent you a LinkedIn connection. I'm playing with John, and John's who we're playing with in PDAC as well.
Okay.
So just as a final point, I think, because we're just way over time, because me and you, Tracey, one of the points I'd make to the researchers, which I really wish somebody had told me before I started on the Merck contracts and patenting, is that if you know that you're going to do this, it might be worth saving up some small stories and side projects to publish during the time that you're trying to get your patents patented and you're under contract to the companies, because that saves you a valley of death where it's horrific, because you know what you're doing, and like UniQuest and the universities will not let you publish until the patents are out. But the other part of the university will sit you in various places saying, where are your publications? Publish or leave. And you're going, but the university says I can't publish. And the university is saying publish or leave. It's a horrendous place to spend your time. So if you're going to start it, save some papers up, even if they're small ones, that you can publish through the valley of death.
I wish you'd given me that advice in 1998.
Right. I wish I had it before I started as well. So this is our job, is to tell people how to avoid the utter pain that we all went through. Yeah.
That's why I started my own biotech, because no one had Fiona to give me that advice, and I was patenting, I wasn't publishing, and I just couldn't get the grants. So I thought, but I had some really interesting stuff that I knew I could translate into man. So that's great advice. Totally support that.
Yeah, so somebody like Tracey doesn't kind of sit down. She just goes and starts her own biotech company.
I have no idea what I was doing. If I did, I probably.
Can I just say for International Women's Day that we've just had, Tracey's a fantastic example of not giving up. I'll hand you over to Irene.
You certainly both are, and we thank you very much. And let's put our virtual hands and real hands together to thank Fiona